So Ben, it's up to you.
No, it's not up to me. It's up to Amélie Gaudin and Kate Scow. That was a nice tribute to Ruben. These two people from UC Davis are going to interact and create a story of soils. And it's interesting. Kate Scow started out as a zoologist, and she's narrowed it down and down and down and down until she's looking at protozoa and bacteria and fungi. Amélie Gaudin is similarly, she's looking at potatoes and rice and almonds and grapes, and she's now looking at roots and the interaction of roots in the soil. And it's great that they're bringing this discussion to avocado growers, because avocado growers have dealt with the high organic matter system for years, and they've been all wrapped up in organic and biodynamic and regenerative agriculture and sustainable and ecological and biological agriculture and permaculture, and they've been fooling around with all kinds of biostimulants. And so we're going to hear from them. So take it away, Amélie and Kate. Thank you.
Good morning, everyone. Kate, we're good to go. All right. So thanks for being here today. Good morning. Kate and I will be talking about healthy soils, what they are, the functions that are of relevance to avocado growers. And we wanted to really dive a little bit deeper today into the biodiversity of soils and how that drives healthy soils. We'll cover how to manage for soil health, what are some of the management principles, how to measure soil health, and we'll apply some of that knowledge to some case studies, especially in almond system, which has been the bulk of my research, and I'll show you some results we've had with various practices. So soil health in general has not really been emphasized in ag in general, and this is especially true in orchard system, where much of the emphasis has been placed on trees and maximizing productivity and input use efficiency. And this has really resulted in some evitable management challenges. We see across the valley problems with compactions, runoff and erosion, some salinity challenges, some soil-borne disease pressure that are limiting yield and creating greater challenge across the landscape. So there's actually a lot of opportunities to build healthy soil in California and elsewhere to support a more regenerative and productive orchard systems. And we often think that, well, soils, what are the impact of soil health on yield? And those relationships are not necessarily very linear. But I want to start with this picture here, which is an almond orchard we started to work at, which has heavy compaction. The grower had deep rib, put some gypsum, work with soil amendments to get rid of the problem. And after a while, the orchard was actually losing productivity. And so we started to work in this almond orchard. And after 2 years of implementing just cover crop and being starting to be mindful about soil and soil health to decrease compaction problems and water infiltration problems, we actually gained in this orchard in year 2 and 3 about 200 to 400 pounds per acre of almond. And we saw that across the valley in various different environments. So there is definitely a potential limitation of yield from non-healthy soils, and putting soil health at the center of conversation can bring some advantages and some benefits to avocado growers. So what is healthy soils? Really, the definition, which is based on the NRCS definition, is really that soil health refers to the capacity of soils to function as a living ecosystem that sustains plants, animals, and humans. And so by this definition, really, soil health term reflect not just the composition of the soil, but goes beyond in looking at the capacity to support agriculture while conserving natural resources. It really emphasizes soils as a living ecosystem that is a very complex matrix that hosts a diversity of organisms, and we'll learn more about that from Kate in a few minutes. So soils are living, they're dynamic, and they support many sustainability and productivity goals. A healthy soil has an adequate fertility, has a good soil structure, has an active and diverse soil community, and help conserve and cycles nutrient and water. And so we can really see linkages with sustainable agriculture in terms of building more water and nutrient use efficient systems, with a potential to decrease externalities in terms of leaching, for instance, decrease in salinity, while enhancing the potential of soils to mitigate climate change. This is the largest sink of carbon we have on our beautiful planet. So soils are also very important for carbon storage, and that carbons also drive soil health. So we have the potential of a win-win situation here. But soil health concept is still nebulous, and it's nebulous because it's a beautiful metaphor. If you think of our health, what is it? It's complex to measure, but really it's a measure of how well a soil can perform its essential function. So much like human health relies on bone structure, respiratory systems, a healthy gut microbiome to digest our food. A healthy soil requires good structure, some good gas exchange, and a healthy living microbial community to cycle nutrients to support production. So it really goes beyond soil composition and describes how well a soil can thrive, but also responds to disturbances and we have a need for diagnostic and measuring improvement in health. And so it's very complex to measure, much like our health. We take a series of indicators. We're going to take the pulse. We're going to do blood work to try to evaluate health and diagnose what might be going on to be able to improve health conditions. So it's complex to measure. It relies on a suite of indicators that can be measured in fields or in the lab. But we try to have indicators that are a little bit more functional and move beyond just compositions of soil. And so as such, the health of a soil is really dynamic. It's dynamic in time, it's dynamic in space across an orchard, in the alleyway compared to the berm, for instance, but also across different soil depths. There's, however, some very key determinant of soil health. Not all soil have the same health potential, and however complicated it might be to ascertain, but there's various determinants. Our health, to continue with the analogy and the metaphor, really is a function of our genes, right? The genes that we receive from both our parents, the environment we live in, and our actions, our diet, our exercise, etc. Soil health, to the same extent, is a function of the parents, so the rocks that have created soils, and so the soil texture is very important. The environment, whether we're talking of an anthropogenic soil or a soil that is agricultural, or more natural areas, whether you're at the top of the slope or at the bottom of the slope. So the environment, the vegetation, the climate is very important determinant of soil health. And then the actions, which are basically growers' management decisions. And so the actions that a grower takes in an orchard impact most of the dynamic properties of soil, especially soil life and organic matter. When we think about we often think of soils across the three somewhat separated dimensions. We think of the soil chemistry, the soil physical attributes, and the soil biology. And when we start to really putting life back at the center, it really allows us to look at this through a more integrated lens. And I like to think about it as an ecosystem where the physical properties we measure is really the habitat for a living and diverse soil community. And the chemistry and nutrients are really thought about as resources, energy and nutrients to support the soil organisms that drive healthy soils. Now on to you, Kate, to learn more about some of those organisms.
Okay, thank you. So I'm going to talk about soil biodiversity, which is the engine or the heartbeat, or whatever metaphor you want to use for soil health, and something that we have been learning about a lot in the last 20, 30 years, and really advancing our understanding of it. And I'm trying to advance. Okay. I'm going to talk from a report that recently came out, hosted by the California Department of Food and Ag on soil biodiversity in California agriculture, looking at framework and indicators for looking at biodiversity within a soil health assessment. And this was 14 scientists throughout the state, including Amelie and soil ecologists, to put together this report that you have access to it there with the code. So soil biodiversity is the primary driver of terrestrial ecosystems, all ecosystems, right? Not just managed ones, obviously. And it's the unseen driver. People think that biodegradation just happens, carbon just cycles, nutrients just cycle, soil structure just comes about, but all these, I think almost all greenhouse gases just happen, right? But all of these particular processes, and most in the soil, are indeed the work, the deeds of soil biodiversity. Microorganisms being a really important part of it, but not all there is to it. And without, something like soil structure, which is seen as just a very physical aspect of soil, is indeed the output of a lot of biological activity. There's this lag. Okay. So soil also provides what we call ecosystem services in the language that is used very commonly today at the global scale. And this is a picture from FAO, Food and Ag Organization, showing that that what soil brings to humans extends way beyond what we see in agricultural systems involved with food and fiber production, but also, in scaling up from the figure that I showed you before, has a lot to do with just the regulation of climate. Carbon sequestration is very much driven by it, but we also have more, things like cultural heritage is like retained and stored in soil. Soil provides building materials. It provides the foundation for things to be built upon it. Water regulation is a huge role of soil. So there's many, many functions that are beyond what we are often, hoping for, anticipating, expecting, managing for in agricultural systems that have much broader impacts on the world when they're scaled up. And agriculture can play a big role in providing some of these services. Sorry, for some reason there's a big lag. So the life that inhabits soil encompasses a vast range of sizes and diversity. And this figure here shows you microorganisms, viruses, even less than that, smaller than that, in sizes at the submicron level, extending up to, what's the visible. Most of the organisms in soil are not visible, but it extends up to aardvarks and armadillos and, very large organisms as well that aren't shown. But this phenomenal amount of diversity that is really quite staggering. And in fact, there's a recent study that was done that quantified diversity throughout the whole planet and estimated that soil is home to approximately 60% of all the biodiversity on the planet. So this is looking at all ecosystems, oceans, and all aquatic systems as well as rock formations and all. And you can see that in particular, this graph is a little bit easier to see. 90% of the fungi on Earth, according to these estimates, live in soil. Encotraeans, which are very small worms, most of them live in soil. About 50% of the bacteria on the planet are found in soil. I mean, obviously plants are definitely predominantly in soil, but just this phenomenal amount of diversity that makes you, number one, I think, appreciate the need to protect soils, to protect this biodiversity, and number two, to maybe try to start to think about how to include it in biodiversity estimates, because most biodiversity estimates are above ground. We don't consider what is living below ground. So soil organisms do not live in isolation. They are in these complex food webs, but they work together in these independent networks. So often the idea of a species of an organism is not very relevant. It's more relevant with pathogens, but often not with these other functions that we're talking about. And you can do these network diagrams for, that are done for power grids and social networks. And this is a soil network showing these just phenomenal amounts of, not just numbers of organisms, but interactions among organisms. And the way we define soil biodiversity is far beyond just body counts, how many. That indeed is important, but there's this definition from the FAO includes from genes to species to communities, alluding back to what I just showed you with those networks, these ecological complexes to which they contribute and belong, and at different scales. And we're going to talk a little bit about how we measure soil biodiversity later on. And management directly influences these intimate relationships between soil biodiversity, shown here, and all the other properties and functions of soil that make up soil health. So these choices that we're making up here, some things we can't control as well. Well, in principle, we could control the climate, but we don't. But management, like in an ag system, is going to affect these interactions between organisms and these other properties, and it's going to impact these functions that that ecosystems provide, the services that then come forth from it, and then ultimately resilience, adaptation, and multifunctionality. And I just want to say one last thing and before I hand over to Amelie, that ag soils are chilly environments for soil biota, often because of the management choices that are made. Not enough carbon goes back in, physical disturbance, long periods of bare soils, maybe not in avocados, but in a lot of other systems. Agrochemical use, fertilizer concentrations being high so that symbionts can't function. And then a lot of practices are for very rapid solutions rather than taking a long-term look at things. Okay, I'm handing it over to Amelie then.
So what are some of the principles to build up soil health and how they apply to orchard systems? When we start thinking of soils and soil health and change over time, change often starts with soil biology and soil ecology and the soil biodiversity. So they're the first responders to different management practices. And over time. So biological activity kicks in as you start shifting your practices. This triggers a shift in nutrient cycling, organic matter accumulation and turnover. And then eventually all of those processes lead to improvement in soil structures and with or water availability. And so there's a progression of visible outcome, and that's why it takes time to build up soil health, because you need to prime this biological activity. And the most degraded it might be, the fastest it might be responding. But that's something that is to be kept in mind. And so there's a lot of invisible and dynamic processes. And really at the center of it is the carbon molecule. It's the primary source of energy to feed this biological activity. And so those carbons come from plants. So plants will fix carbon dioxide from the atmosphere through photosynthesis and accumulate simple sugars, mainly glucose, in the stem, leaves, and roots. Some of it is translocated directly during plant growth back to the soil microbe systems via exudates, and a lot of it actually come back through residue. There's also the carbon from plants, and there's also the carbon that is in soils as a source of energy and nutrients for the microbes. And that carbon might be dissolved or in aggregates and in various different forms. So the goal really, when we start to take a soil health building lens to management, is to think about accumulating the carbon stock or the carbon, if you think of a bank account, right? And you want to build your funds, your stocks, you have various solutions, right? You can earn more, fix more carbon and deposit more into that account. You can lose less or withdraw less. And you can also prioritize some long-term storage. Well, the same ideas are in play for carbon. And so to increase inputs, really, it's about having a steady food supply for microbes and the soil food web. So the management emphasis should be around having a diversity of carbon input in terms of forms, but also timing. Carbon that is going to be released in different some slow release of a variety of organic compounds over the course of the year. So we think about amendments, having living roots, residues, grazing can be a potential practice to diversify the type of carbons and microbes that are in the systems. And realizing as well that not all carbon inputs are equals. Microbes need a balanced diet. Carbon is the energy, but they also need other nutrients to build cells. So nitrogen, phosphorus, potassium, sulfur, all of this needs to be in balance for the soil biology to kick in and for carbon to accumulate in soils. In terms of decreasing losses, a lot of it has to do with disturbances that break aggregates where carbon is actually stored, decreasing the impact of disturbances on the habitat. For instance, for fungal symbionts that really rely on hyphae, etc., to exist and communicate. And so any type of disturbance, such as tillage, for instance, will disturb those networks. And that's where we have a real advantage in perennial systems. We definitely have less tillage and soil disturbance than in annual systems. So a real potential to really look deeply at soil health in perennial orchard systems. There's some level of chemical disturbance. Chemical disturbance is a disturbance. And also thinking of decreasing losses by having carbon incorporation into deeper soil layers, which is something that might not be happening so much in orchard system compared to annual system. So really, to build up carbon, it's about increasing input, decreasing losses, and locking on this carbon into soils for the long term. How do we do that? Well, there's maintaining living roots in the soils, keeping soil cover at least a minimum of 30% soil cover 100% of the time. have frequent and diverse input of organic matters from different sources. And although agriculture, we're in the business of disturbances, so it's hard to not disturb a soil, but trying to think through ways to be more strategic about disturbances. Eventually, all those principles are realized through maximizing biodiversity in the system. And not all practices can be applied to the same extent everywhere, just because it needs to be adapted to the landscape and to the communities. For instance, if you're down south and where you have way less rainfall than us here in Yolo County, you might not have the same tools in your toolbox. And that's why it's helpful to think through those principles, because they can really be adapted to your resources, the landscape, and some other determinants of the possible. So those are the principles for management, but it's also very important to keep track of where you are in terms of soil health and where this is going. And so there's various measures of soil health and indicators that we use.
Okay, great. So I'm going to take over here and talk about indicators and interpretation. So many of you, I'm sure, are familiar with these typical soil fertility tests that focus primarily on nutrients in the soil and pH, and maybe sometimes texture will be thrown in, sometimes cation exchange capacity. But as Amelie was talking about in soil health, it's this intersection of chemistry, biology, and physical properties. And these chemistry tests, the typical soil fertility tests, just scratch at the surface of what is a much bigger, more complicated pool of chemicals, a much more complex chemistry going on in the soil, a lot of it important in soil organic matter. So what do we want? And so when you want to decide what to measure to get an idea of soil health, sometimes people think that's just a ready-made thing. Oh, soil health, it's like with humans, it's temperature and blood pressure and boom, that's your soil health. That's your health assessment. And those, of course, are used for humans as just sort of the front end in triaging problems, and you'll go deeper to explore what is going on. And same with soil health. What aspect of soil health are you interested in? And I think we've sort of oversimplified soil health assessment by not thinking a little bit harder about what it is we are interested in. So here are characteristics of a healthy soil, and this is from the Cornell Soil Health Assessment that I'm going to talk about a little bit. But we're looking for till, sufficient depth, enough, but not too much, sort of like Goldilocks, supply of nutrients for plants, small populations of pathogens and insect pests because we can't get rid of them entirely, good water storage, good drainage, large populations of beneficial organisms, low weed pressure, free of harmful chemicals and toxins for not only plants, but also microbes and also people who work in the systems, and then an overall resistance and resilience to degradation and perturbation. These are things that we're looking for. Now, if you look at those properties, some of them we can measure directly, but a lot of them we're going to measure using indicators to kind of get at them, but we can't actually determine them directly. Now, there's a variety of different kinds of soil health assessment tools that have been developed for different users and different purposes, from simpler like scorecards that have observable indicators that can be on a checklist, and a farmer can walk around and look at different aspects of the field and come up with an assessment that's really valuable because it's on the ground about soil health. There's some kits that have been put together that can be used by growers or extension specialists to go in and do in the field simple analyses. Then there's lab-based assessments that are require more specialized equipment and more precise measurements. And there's several examples of that. And we're going to talk more in detail about these. There's landscape level assessments as well to scale up. And then there's tools that, Amelie is familiar with, looking at aspects of multifunctionality and resilience that have to do also with soil health. But we're going to focus on this intermediary approach. And I'm going to focus on the Cornell Soil Health Assessment Program. This is called CASH, Comprehensive Assessment of Soil Health. Cornell has done the most work anywhere on trying to understand what are the components of soil health, doing a lot of research to go along with it, developing soil tests, and then putting together this framework that you can use to get sort of a more comprehensive view of the health of your soil. And also they provide a service and you can send samples there. But just, I did my PhD at Cornell, and I was working with a soil that had like 4% organic matter, 5% organic matter. And I come to California and I see these soils that just have 2% organic matter. And I was thinking, oh my God, this is what a terrible situation California is in. It's got such low organic matter soils, and just, giving you a taste of the differences between what soils are like in the Northeast United States and what has been developed through this Cornell assessment, which is for soils throughout the Northeast. Some of it's not going to be completely relevant for out here in California or other regions, but I focus on this because they have done the most work. It's very impressive and provides a model. So in the Cornell assessment, how is healthy soil measured? They look at different particular indicators. So for chemistry, pH, and then phosphorus, potassium, magnesium, zinc. They screened like a number, like 59 different indicators, and did a lot of work on what seemed to be best in discriminating different kinds of soils. And they came up with this smaller set, which is part of their assessment. Biological characteristics, soil organic matter, soil protein, respiration, and active carbon, and then physical properties, texture, water capacity, aggregate stability, and a measure of compaction. And to be able to interpret it, once they go in and measure a particular parameter, for example, soil organic matter content, you need to be able to interpret it. Like, what does it mean? It's one thing to have it. It's one thing to compare maybe two of your soils against each other. But what if you want to know, what does that mean? Is that a healthy value, or is it, an unhealthy value? And so they did a lot of work on developing these scoring functions for these different soil health parameters, from low to optimum to high. And this is an example of just one of these functions. This is one where high is good, green, and low is not as healthy, and that's red. But there's also other parameters that go in the opposite direction, right? It'll go from high is going to be bad and low is going to be better. And there's also ones that are normal distribution kind of functions where there's a midpoint that's the highest. So just letting you know that there's other of these curves, scaling curves, that have been developed for different parameters. And this scaling was developed by collecting soils throughout the whole region of the Northeast to build a database and then be able to look at the distribution of these values across different soils and come up with this scaling. So their reference database is relevant to the Northeast, right? Again, I'm underscoring that the scoring may not be as relevant for California. Organic matter, again, would be an example. They say our soil is unhealthy based on organic matter if you just pop it into this. And here's an example, and this is a little bit detailed, but it shows you what you get. Here are the physical properties of the soil, the chemical one, I'm sorry, the biological ones, and then the chemical ones. Here's the number that they get when they measure it. So they've got the list of protocols that they use for coming up with these different numbers, like for these different parameters, the different indicators. So here you have the scale, these direct values, and then here is the rating based on the scaling that I just talked about. And in this soil, a lot of like the chemistry was looking really good, and so was the water capacity, but aggregate stability was really, really poor. So got a very low score. And there's also an overall quality scale that is a composite of all these different individual scores, which I have a lot of skepticism about those kinds of composite scores, but they do estimate it. And then here, in the soil health assessment method, they they will call out aggregate stability as being low, and then here are management suggestions for these constraints, like what are things that you could do physically and biologically to help address that. So this just gives you the inside, the guts of a soil health assessment. So again, I just want to underscore that this particular framework and other ones as well, most of them have been developed more for the Midwest and Northeast. And there's obviously big differences in climate and farming systems, types of soil, operational constraints. We have to irrigate, and that is really different from places that are dependent on rain, and there's plenty of it. And so it can limit what we can do here in terms of some of those values. And we also have a phenomenal amount of diversity in our agricultural systems. So this reference values, right, is something that is lacking. There's work going on in California about this, and also in Washington State. I think there's been a lot of work in trying to calibrate this sort of framework, or modify it to work for our regions. Other considerations, in doing these measurements of for soil health are, the sampling design. There's a lot of information in the Cornell handbook, and a manual that gives a lot of guidance on how to go about. When do you sample? Do you sample in the when things are very quiet, like in the fall before the rains come, or do you sample when there is rain coming into systems? Do you sample in the middle of the growing season? There's you're going to get very different for some, especially the biological parameters. You're going to get very different measurements based on where it is. Where do you sample? I mean, do you sample randomly and try to capture all the variation in an area, or do you target particular areas in close to the tree, or in the furrow, what do you call, alley, in between, depending on the question. Depth matters. A lot of the soil measurements are done in the top 15 to 30 centimeters of the soil. That might be important for some measurements, but it's going to really miss some important things going on deeper in the soil, particularly with respect to carbon, where there may be a lot of storage of carbon below the area that you're looking at. And as we go forward, the indicators continue to evolve and be developed, and we're really seeing that with soil carbon total organic carbon is, sure, it's still important, but there's a lot of other pools of carbon that say a lot more about what is going on in the system, and is it moving in a direction of perhaps having net carbon sequestration, or also what is the kind of carbon? Is it good carbon for microorganisms and energy? So there's a lot of work and a lot of new indicators coming on, and this is particularly with the biodiversity indicators. So I want to say that there's other soil health frameworks and tools. I don't think that any of them are quite as usable directly as this, except maybe the Haney test, but they a lot of information and data and protocols and recommendations at the Soil Health Institute. NRCS has a soil health assessment, protocols and recommendations, and then the Haney soil test is popular, used a lot, which is a much smaller scale test that doesn't involve as many different measurements in the lab, that includes some of the parameters that we are talking about. And there's other ones. Solvita, I think, is, again, they only look at part of what we've been talking about. I guess, Amelie, I'll just briefly talk about the soil biodiversity indicators. Yeah, I think, yeah, it kind of fits here. So going back to the report that I talked about at the beginning, that CDFA hosted, the below ground biodiversity report, we did a lot of work on soil biodiversity indicators. So you saw that what was in the Cornell test were not direct measurements of organisms, but measurements of the deeds of the organism. So like process rates and products that come from biological activity, but not directly measuring the organism. In our report for the CDFA, we looked at a lot of what would be valuable to measure if you really wanted to drill in on soil biodiversity more. And so we grouped the different kinds of categories or types of soil biodiversity indicators into these five groups. So one would be abundance, meaning like how many organisms are present. So this could be a total microbial biomass, which is everybody together, not caring who they are. But there's also now the ability to target particular groups of organisms. Like if you wanted to know about a pathogen, or if you wanted to know about an organism that produces nitrate, so specific functions. I mean, I'm sorry. It's kind of going into identity. So identity, who are they? But you can also, with some of the molecular tools, find out how many there are of particular organisms that you identify. So a lot of the new work on determining who the organisms are is based on DNA sequencing. And these tools are out there expanding. There's lots of companies that will sell you analyses doing sequencing of soil organisms, and there's a lot of excitement in being able to learn who is there. But I have to say that it's often difficult to interpret it once you find it, because there's this phenomenal amount of diversity, as we talked about before. And this is an area of a lot of research. Then there's functional traits. Remember, in that definition of soil biodiversity, it isn't just who you are. It's not just body counts and identifying, who in particular you are. It's what you do. And often what they do is the collective action of multiple organisms. So you're not necessarily looking at what one organism is contributing with its function, but you're looking at this capacity of the soil to do work, to carry out some of these these functions. And again, there's molecular tools that allow us to go and look at that. They're not, I don't think so commercially available. And again, there's a lot of research that still has to happen. Then another important thing that we look at is interactions. And this is even further out. It says that network picture that I showed you is one way to get at what are the interactions of the organisms within your communities. And this involves a lot of modeling and big data. And it's not something that I think we're routinely doing in agriculture yet. And then processes. And this is something that we're more familiar with, which is also in the Cornell test. It would be like respiration is a process. So it's measuring the bulk result of the activity of all the organisms in the soil. It could be denitrification, any sort of greenhouse gas. Most of them are organismally produced. So like, methane generation, or the other way, how much carbon is going into the soil. So these are also other things that can be measured. All right, and then here are some of the methods. Yeah, it's kind of repeating a little bit of what I said for these different categories, what you might use from simple fumigation extraction. PLFA, phospholipid fatty acid, is used a lot for looking at biomass. Identity, the DNA. Morphology for nematodes. Functions, that's getting into metagenomics, metaproteomics. Yeah, interactions, and then processes where you can actually look at, as I said, what are the deeds? What are they doing? Which is often more important than who they are. So which indicators do you choose? Like, there's phenomenal amount, right? And which ones do you use to represent what you're interested in your soil? And in this report, we said it depends. It sounds like a scientist, doesn't it? So, but we developed 4 scenarios or use cases to illustrate how you might use biodiversity indicators. So let's say you wanted to know what's the status of the soil biodiversity across the state of California, which is essentially developing a database. So that would be perhaps sequencing DNA. Another thing you might want to do is the California Healthy Soil Program having benefits for soil biodiversity. So that would be a different set of indicators. And this is all in the report. The third one that might be more relevant to you is how can you use soil biodiversity indicators to help guide particular decisions to go after particular needs, like disease suppression or reducing nitrogen loss. And then there's this fourth kind, even though they're all number ones, which is what can you do to engage the general public about the importance of soil biodiversity? And there you might bury underwear. I don't know if you've heard about that as a method, but there's some more popular ways to engage people to see what are the benefits of soil biodiversity and what is a healthier soil than a less healthy soil by looking at processes like that. And here's an example of like if you want to know how to manage nitrogen more efficiently in your soil, and your goal would be to adjust end inputs. The audience would be the growers, but also land managers. So there you might measure populations of nitrifiers and denitrifiers, nitrogen cycling organisms, microbial biomass in general, and these fluxes of N, these changes in these process rates, just as an example. Okay, and I just want to call out one thing before I hand it over to Amelie, and that there's another recent report that has come out, which is National Academy Science, Engineering, and Medicine that focused on the linkages between soil health and human health with these chapters here. And it's just so you know that it exists. A lot of the focus is on agriculture and more information about ag management, but also looking at the interconnection between humans and soil via the microbiome. So starting to look at what are those interactions, and some through the food systems. So if you have an interest, that's a report to check out. Okay, hand over to Amelie.
Right. So there's a large varieties of indicators that can be used. And I wanted to show you a little bit of some case studies as to how we've used those indicators to measure impact of different practices on soil health metrics, and basically how healthy can we get. Even though putting a number on it might be difficult, we wanted to see kind of what is the range in terms of soil health in orchard in a definite environment. And we actually have a lot to build on. There's many systems that growers have been innovating with, and advisors, and growers have been really forward-looking into implementing a series of practices that aim at building this carbon stock and improving soil health and soil biodiversity to support agriculture. So some of the practices that we're talking about go from the habitat from around the orchard, such as peel margins, to something more in situ, such as whole orchard recycling, having organic amendments and compost, thinking about cover crops and understories, cheap mulches, hulls and shells and biochars, and I work a lot on grazing in orchards. So one of my favorite soil health practices as well. So what we decided to do is to kind of move away from those trials, use replicated trial, and kind of embrace all the innovations growers have been playing with, and measure soil health across various orchards that have had different application of the principles I talked about, and some of the practices listed here. I want to start with whole orchard recycling. That's a practice that does not necessarily require a big shift in the system, and really it's about setting your system for success in terms of soil health. So when you do whole orchard recycling, that's pre-plant. When you're going to turn over an orchard, for instance, you're going to chip the trees and incorporate them into the soil before replanting new trees. Those are from almond systems that actually never worked in avocados, but I wanted to show you some results, and there might be some avocado specific concerns here. But what we find, we went to measure the impact of whole orchard recycling on soil health across various orchards that were recycled between 9 and 10 years ago. And we compared it to orchards that weren't recycled, where residues were burned or exported. We found a 32% increase in the ability of soil to store water. So we, you've got 30 to 60 tons of carbon coming back in from those trees. And so a very important practice, very impactful practice in terms of long-term soil health. So greater water holding capacity, that's one of the few practices we see that really get your soil to those water storage. 32%, which is actually pretty significant. We see twofold increase in infiltration, 14% less compaction, better aggregation. And all of this resulted in when you implement deficit irrigation, or when water is scarce and you got to cut down on irrigation, we see that under water stress, the trees are actually more resilient. They're able to maintain a higher water status and water use efficiency when from using this larger water storage in the soil. We see 20% yield increase when water is scarce in almond systems during hull splits. So a beneficial practice here to build up resilience to this very uncertain water supply situation that we have across the state. And we see 10 years after recycling the orchard, 52% reduction in nitrate leaching. So we have a system that is storing nitrogen in an organic form, that is cycling nitrogen. That's what we call a tight nitrogen cycle, where nitrogen is made available over time without having this accumulation of fast-moving nitrate molecules that can leach into water bodies. We see no yield trade-off There's a little bit of a tweak in the fertilization of young trees just after recycling, because you have all this carbon, and so in my the soil is a little bit unbalanced, and the microbes also need nitrogen. So you have to up your nitrogen fertilization to young trees for a year or two, but then can resume normal fertilization guidelines. And we've seen, at least in almond, no replant disease or low pest and disease potential. This is in almond. It's not necessarily the same situation in walnuts or in other crops with higher tannins. So whole orchard recycling, really, it's setting up your orchard for success, and investing in soil health pre-plant, so that it carries over the lifespan of the orchard. If you're interested, we have some guidelines that we've put together, a guide for growers and a website to learn how to recycle and what are the potential benefits and things to watch out for. The second strategies that we've collectively looked at are compose, hulls, and shell amendments. And the idea here, as at least in almond, we're moving more and more toward off-ground harvest, is to recreate this kind of forest layers that are really cool in avocado system. We see 2 or 3 years after the first amendments, a short-term increase in soil fertility. Hulls and shells can provide about 80% of the potassium requirement of an almond tree. So very high potassium levels. We've got so a very relevant source of nutrients. It does increase soil organic carbon, especially in the topsoil layers. And so there's a bunch of associated benefits with that in terms of cation exchange capacity, etc. We definitely see an up in biological activity and a mulch effect for hulls and shells, so lower soil evaporation and higher water infiltration. So some good practices as well for building up soil health, and some of the recommendations for compost and hulls and shell amendments have also been integrated into some best management practice guide through a nitrogen management lens. Winter planted cover crop. We've developed also a best management practice guide to help grower implement cover crops 4 years after implementation of cover crop compared to bare soil. We definitely see a win in terms of water infiltration, aggregation, and compaction. There's more biological activity. It's sometimes cover crop alone. It will be very difficult to get to those increases in soil organic matter that might bring the soil to additional benefits when it comes to water retention, etc. Cover crops really are often not enough to get our soils to the highest health potential. But they have many co-benefits in terms of fostering of pollinators, helping suppress weed, and also reducing emergence of some pests and disease in almond systems. So it's not just about soil health, at least for cover crops. And really the real win comes when we start stacking practices. I wanted to show you in the last 5 minutes what can be some of the outcome. We looked at 12 almond orchards across a gradient of management on a similar soil texture. So we took a transect of 100 kilometers and just stayed in there. And so all the soils are a similar texture. And we had various groups of orchard, one that has bare soils and some sporadic inputs of compost and almonds, hulls, and shells. Some group of orchards that had living covered, so planted cover crops or resident vegetation in the winter or continuing well into the summer as living plants or residue mulches. And then the third group where we wanted to really see what can we obtain from having higher echelon of biodiversity there and integrating an animal to graze those cover crops, whether in the winter or in more continuous living pastures. I'm going to try to make a long story short here, and I'm not going to show you all the indicators we looked at. I just chose a few, but So this is the group A, this is the group B, and this is the group C. And the colors here indicate whether those numbers are significantly different. And so what we see in terms of lowering compaction, for instance, we see a very large potential of reducing, well, there's potential to reduce compaction, even though you have an animal in the system, if it's well managed. So having those living groups in the system is actually super important to lower compaction. In terms of soil carbon, you can see that we can achieve more than double the amount of soil organic matter when we start having and all those sorts, sorry, I forgot to mention, the practices were implemented at least for 5 years. But you can more than double your total carbon when you start stacking those practices and having various management principles being implemented. The microbes are really jazzed up, they're respiring, they're very active. A lot of increases in the soil protein, which means some of the body and the carcasses of the microbes. Higher total nitrogen and cycling of nitrogen, and you can see that you can have all of those benefits and not necessarily improve water holding capacity. And so we need to make sure that when we claim soil health improvements, we have much more research to do in terms of this relationship between soil health and water, and how that trickles down to saving inputs. So there's a large untapped potential that can bring benefits. I think it's important to remember as well that those soil health building practices also have other co-benefits to tackle some additional sustainability and productivity challenges when it comes to weed suppression, pollinator conservation, pest and disease regulation, and keeping nitrogen in the systems, for instance. So in conclusion, we have large potential in orchard systems. We're measuring high levels of improvements when you start stacking practices. What's cool about orchards is that you have multiple designs that you can implement in space and time to implement those practices. And we've seen very little yield lag, at least in almond and walnut systems compared to annual systems when implementing some of those practices. It can be very aggressive, trunk to trunk, or it can be something a little bit more conservative as we start learning about how to implement this in different environments. There's, however, a lot of unknown, and we need to realize that we've been working on soil health for the last 6 years. And so we've got a lot of ground to cover in linking soil health and biodiversity, but also making this link to potentially be able to cut on inputs and having higher sustainability metrics. Implementation can be a challenge. We need more technical assistance in terms of helping grower implement some of those practices, equipment sharing frameworks, and having a flexibility and exchange between growers is going to be very important. It's not a silver bullet, so each action is going to be context specific. There's a value in experimenting together. I mean researchers and growers researching locally and on farm to develop adapted management guidelines, and there's often a cost associated with building soil health, and so we have incentive at the state levels, but we see increased motivations from growers to try to shift the paradigm and the narrative for management. And so there's a large potential to realize the potential of soil health in avocado orchards. With that, Kate and I thank you for your attention and for sticking with us for an hour. And we're happy to answer any thoughts or questions you might have.